That was the research group. So we're going to get into the talk at hand. I'm not going to make any reference to God, any reference to intelligent designer, or anything. I'm just going to allow science to critique the science. What do we have to say about origin of life? What does science tell us? What can we infer here? And so by choice, I am not going to mention God, because if I mention God and Jesus Christ, people will say, oh, Tour introduced the baby Jesus to talk about this. It's not a real talk. All right, this is pure scientific talk. This would work in any medical school, in any chemistry department. Next slide. So this is a cell. What is the origin of life? How do you get a cell like this? What is the origin of something like this? This is an amazing machine. A cell is an amazing machine. It's not just a blob of protoplasm. Every day it gets harder to have the origin of life, to come up with a scenario, because the origin of life becomes more and more complex every day. A cell is a factory. It has the lipid bilayer, which is extremely selective to let certain things in and not other things. It has all of this substructure in here, these little areas where energy is made in here. It has these microtubules which can form so you can move matter from point A to point B. If you go to a factory, what you see is these overhead hanging machines that are moving materials from point A to point B. In these systems. And the way they do this is they build these racks. But same thing happens in a cell. You get these microtubules to move material from point A to point B. And then as soon as the material is done moving, the microtubule breaks down and then assembles some other place. You say, well, why doesn't it leave it there? Because then the cell would become too rigid. So it just rebuilds it. It's just amazing factory what's happening in a cell. This is what we have to make. If we want to have origin of life, you got to start here. You don't start here. You start with a single cell. Just build a single cell. That's what we have to do in origin of life. Nobody has ever done this. If you've been taught that simple forms of life have been made, that is a lie that you are believing. Somebody told you a lie. That has never been done. Next slide. Organisms care about life. Molecules don't care. Chemistry, on the contrary, is utterly indifferent to life. Without a biologically derived entity acting upon them, molecules have never been shown to evolve toward life. Never. Molecules don't care about life. They don't know anything about moving toward life. They have no brain. Organisms want to move toward life and keep life going. Molecules don't care about life. Nobody has ever seen molecules assemble toward life. Never. It doesn't happen without a biological entity working on them. I asked all my colleagues, can you show me an example of this? Of molecules moving toward order, moving toward an ordered system where you have a non-regular assembly. Regular assembly is like A, or A, B. That you can get pure thermodynamic assembly. But non-ordered assembly is a non-regular pattern. That's what you have in DNA. We know from computer science you have to have non-regular patterns in order to have complexity for living systems. And I asked my colleagues, do you have any example of molecules without a biological entity acting upon them, moving to give an ordered assembly that is a non-regular pattern? And they sent me papers where chemists have taken molecules and assembled them in that way. I said, non-biological entity. You can't have a human doing this. Molecules don't move toward life. Well, even if you want to have molecules move toward life and you have human beings working upon them, can the human beings do it? And the answer is no. Next slide. So almost every chemical synthesis experiment in origin of life research can be summed up by a protocol analogous to this. They purchase chemicals, generally in high purity, from a chemical company. All right, so that's what they do. They first purchase the chemicals. They mix those chemicals together in water in high concentrations in a specific order under some set of carefully devised conditions in a modern lab. Then they can obtain a mixture of compounds that have a resemblance to one or more of the basic 4 classes of chemicals needed for life. So what you need for life is you need carbohydrates, nucleic acids, amino acids, and lipids. That's what we know. All life we know composes those 4 building blocks. So they try to make those 4 building blocks. Then they publish a paper making bold assertions about origin of life from these functionless crude mixtures of stereochemically scrambled intermediates, much like Miller did in 1952. Nothing has changed in 66 years. Nothing. Nothing's changed. Exactly where we were in 1952 is exactly where we have remained. Think about what's been done in science in the past 60 years. Think about how we have now satellite connectivity. Remember, in 1952, there had never been a satellite. Now we have satellite connectivity. We have cell phones. We have structure of DNA. We can manipulate DNA structure. Nothing has changed in origin of life studies in 66 years. That's important to realize. Then you engage with the ever gullible press to dial up the knob of unjustified extrapolations. Watch the mesmerized layperson exclaim, "You see, scientists understand how life formed." Then you encourage a generation of science textbook writers to make colorful, deceptive cartoons of raw chemicals assembling from cells which then emerge as slithering creatures from a prehistoric pond. That is exactly what is done every one of their experiments. It's all done like this. Every one of their experiments can be fit into this. And so what's going to happen is I'm going to go back and a bunch of you are going to send me articles. Look at this. These people have made life, haven't they? It fits into this. Trust me, it fits into this. Every one of them. Next slide. So here's the synthesis problem. If you just want to make the molecules, remember we have to make those 4 classes of molecules. If you just want to make the molecules, here's what you got to do. Molecules that compose living systems almost always show homochirality, meaning that they have one-handedness, not the other. The vast majority of biological molecules, except for very small ones like water and acetic acid, anything larger than that, they're mirror images. Just like your left hand and your right hand are mirror images. They're non-superimposable. You can't put a left-handed glove on your right hand. It doesn't fit. All molecules are like that in biology. That is hard to make. It is very hard to make just one mirror image of a compound. It can be done, but it's very hard. So that's part of the problem. When you're building molecular systems, constant redesigns are needed to take the synthesis back to step 1. So that take the synthesis back to step 1. So in other words, when you're going and you're making something, you're like, oh boy, those conditions didn't work. My stuff decomposed. So you go back to step 1 and you bring through more starting material. And so you take small amounts. You try this, you try this. Anything that would be happening in a prebiotic earth, it's marching along trying to make something. If it makes a mistake, you can't pull that entity off. Very hard to pull the entity off. Once it's on there, it's on there. So you've been going along, say, for 400 million years, if you want to take these sorts of numbers, and all of a sudden it's put a wrong moiety on there. Uh-oh, what am I going to do? Well, you got to go back to step 1. I got to go back 400 million years? Yeah, you got to go back there. Well, I don't know how to go back. Well, why not? Because I never kept a laboratory notebook. When you don't keep a record of it, you don't even know how to go back. You don't even know what you're going toward. Because it doesn't know that it's moving toward life. Remember, molecules have no brain. They don't know where they're going. That's the synthesis problem. And they don't know how to stop the course of progression or why to stop. There's no target. They don't know, I think we'll form life today. I think we'll make a certain cell. You don't know that. Molecules don't know that. So they're going along and chemical reactions are happening. It doesn't have a target. When you're in the lab, you're going toward a particular target. You know where you're headed. Here it doesn't know when to stop or how to stop. Maybe it's made a carbohydrate. It doesn't say, well, I made a carbohydrate. I think I'll stop synthesizing. No, it'll go on and make something else from that. It doesn't know how to stop. Time, although claimed to be the great savior of abiogenesis, that's before there's biology, abiogenesis can actually be the enemy. For example, carbohydrates are kinetic products. They undergo caramelization or the Cannizzaro reaction. They decompose. So in other words, when you make a carbohydrate, that is not the final product that would form in that reaction mixture. You have to stop the reaction. So a chemist watches the reaction and they stop it at a certain time to stop that progression of the molecule going on. If this is just undirected, unguided, it keeps going to other garbage. Carbohydrates are kinetic products, meaning that they caramelize, they polymerize into a bunch of trash. Just like when you take sugar and you heat the thing up on the oven and things turns into caramel, that's what happens to carbohydrates. They don't stay nice, simple carbohydrates. They end up actually dehydrating. A prebiotic system doesn't have the ability to easily purify the structures. You have to be able to purify, because if you cannot purify, then the byproducts build up in the system and they start using up your starting material and they start inhibiting the reactions that you want. You have to be able to do purification. Without purification, you can't work. Every chemist has to run a reaction, then you stop, you purify, you get it pure, and you go on to the next step. Once in a while, if it's a really pure reaction, you can go on 1 or 2 steps, but you have to purify. Reagent order is essential, reagent addition order. You can't just say all the reagents mixed together and you get what you got. You're making a cake, you got your flour, you got your milk, you got your eggs. You say, well, I think I'll just add the frosting now. No, there's an order to this. This is real. This is what you do in chemistry. Things have to be added at certain times. You can't just add it whenever you want to. Reagent addition order at proper timing is critical. The parameters for temperature, pressure, solvent, light, no light, pH, atmospheric gases have to be carefully controlled in order to build complex molecular structure. There's no way around it. This is what's needed. You have to have characterization at each step. This is hard to do. Chemists have to characterize things at each step because you have to make sure what you've got before you can go on to the next step. How does nature characterize? Well, right now, biological systems characterize things by every time it makes something, there's an enzyme that checks that structure. If it's not the right structure, there are other enzymes that come and chop that up into smaller pieces and try rebuilding it again. You get a mistake in the DNA. You have enzymes that run up and down the DNA, find this mistake, excise it, and then stick in the right base there. But in a pre-biological world, there are no enzymes. How does it check it? And every time, whatever it's using to check it, whatever system is needed to check it is more complex than the system that it's checking. So where'd you get that from? Nobody knows. Everybody's clueless on this, but nobody wants to admit it. There's the mass transfer problem. This seems like not a problem to you, but let me tell you something. This is the killer of it all. Are there any synthetic organic chemists in here? Any synthetic chemists here? Any synthetic chemists? Get your hand up high. One there. Okay. So and one there. Okay. If I say something that's not true, I want you to stand up and just say liar. All right? If I say something that's not true, just stand up. All right? Because I want these people to know, for all they know, I'm lying. You have to verify on this. All right, the mass transfer problem. Anybody who's done complex organic synthesis, what you do is you start with like a half a kilo of material and you go along and your yields are never 100%, you're purifying, you're going, and you end up with 2 milligrams and you're not at your target yet. So what do? Then you go back to the beginning and you make more and you follow the defined route that you had defined before, and so now you can keep your yields higher, but you keep having to go back and pull up starting material from the rear. You're bringing up more material. How do you do that? So say in nature they started, say somehow it started and it got a kilo of formaldehyde and acetone, and it's going to start combining these. So these are going to go along and start reacting. At some point it's going to run out of material. How do you go back to the beginning and get more? Again, you never kept a laboratory notebook, so you never know how you got there. So even if you could repeat it, go down the same junky path and you'd run out again. You'd never get to where you want to go. Next slide. This was the first generation motor that we put in the car. It worked, but it only rotated at 1.8 revolutions per hour. When we rebuild it without that sulfur atom there, then that 5-member ring, then it goes 3 million rotations per second. All right, so small changes can make a big difference. Well, what do? Well, what you is you just come here and you erase that sulfur and you do that. Now that you can do on a blackboard. But this took us back to step 1. This is what I'm talking about. When something, you're going along and you want to build and you say, oh, I'm just near life. If you have one thing wrong, it doesn't work. So, well, you just modify this and it'll work. All right, go back to the beginning. Go back 1 billion years to the beginning. It's too hard. Nobody knows how this thing is solved. You can't build the molecules. Next slide. And then, so remember, these just to make the molecules, the simple molecules, you got to have the carbohydrates, you have to have the lipids, you have to have the nucleic acids, you have to have the proteins. Even if you make an amino acid, how do you hook the amino acids together? Amino acids don't hook together by themselves. We're not even talking about order. How do you just get them to hook? I mean, somebody sent me a paper just last night. I mean, here's a paper just came out on self-folding molecules. Well, duh, that doesn't prove anything. You build a molecule, it folds up. Big deal. How do you get these things to hook together in the first place? Nobody knows. That's with amino acids. How about with nucleotides? You want to get these things to hook up. People will make you see, we got a nucleotide. We got DNA. No, you don't have DNA. You got to get these nucleotides to hook up. Look at a DNA synthesizer. How many steps of blocking, protection, deprotection you need to do the complex chemistry just to get one linkage made? Nobody knows how. You can throw all these together in a flash. People say, well, the concentration of nucleotides was very high. I've read this in high school books. It was very high. And these things came together. You calculate the concentration, the world would have to be raining nucleotides. And even if it were raining nucleotides at those concentrations, they wouldn't hook up. They don't hook up. You need enzymes to hook them up. But this is prebiotic. There's no enzymes here. Or you need very complex DNA synthesizers. Those weren't there either on early Earth. Now you have to assemble it. So say we had the molecules, how can you get them to assemble? So here's the assembly experiments. A protocell is a self-organized, endogenously ordered spherical collection of lipids proposed as a stepping stone to origin of life. So that's what a protocell is. But most so-called protocell assembly experiments in origin of life research can be summed up by a protocol analogous to this. You purchase homochiral diacetyl lipids from a chemical company, or you synthesize scrambled lipids from smaller molecules. Add those lipids to water and observe a small amount of it to form simple and expected thermodynamically driven assembly of those lipids into synthetic bilayer vesicles upon agitation. Sometimes the researchers will add other molecules that get engulfed by those vesicles. Then you publish a paper claiming that the synthetic vesicle is a protocell and suggestive of early forms of life. Engage with the media, hype it up, watch the layperson be misled. Every assembly experiment, I'm telling you, can be summarized in this. Whatever you send me, don't send me anymore. I've dealt with it all right? I get enough of them. And people say, well, what about this one? What about this one? And it fits all into this. It's all garbage, all garbage. Next slide. Next slide. Okay, so the complex cellular membrane. So this is actually what the membrane of a cell looks like when it's made really, really simple in cartoon form. So researchers have identified thousands, actually 40,000 different lipids now have been identified in cell membranes. Not just one. Every protocell experiment, they use one type of lipid. Actually, a cell is made up of thousands of different types. Mixtures of monoacyl lipids destabilize the system. So when they have their monoacyl lipids, those would destabilize the systems. You can't use those. Lipid bilayers surround subcellular organelles such as nuclei and mitochondria, which are themselves microsystem assemblies. Each of these has their own lipid composition. So this is a lipid bilayer. You have some pointing out toward water, some pointing in toward the water on the interior of the cell. And the outside ones are different than the inside ones. Nobody knows how that was done. Nobody knows. Clueless. Nobody knows how that was done. Every protocell experiment just uses homogeneity throughout the whole thing. And so it doesn't, it's not really a protocell. It's not really reminiscent of a cell. Now, within each one of those, you have other organelles like nuclei and mitochondria that have their own bilayer assemblies with their own constitution. Different than what's on the outside of the cell. Nobody knows how that was done. Plus, these have these proteins. So there's this non-symmetric distribution. Then there's proteins that go through here. These are ionophores. That allows certain things into the cell and certain things out. Without that, you can't let anything into the cell. It's going to kill the cell. It has very discrete sensors that allow certain molecules to get in, certain other molecules to get out. It has ionophores that allow certain ions in, certain ions out to keep the ionic concentration at a certain level for maintenance of that cell. As soon as that ionic concentration gets off, you know what happens? Boom! The cell explodes because the ionic concentration has gotten off. It blebs and explodes. Nobody knows how that is done. None of the protocell experiments, none of them have these proteins going through them as these control gateways. Then there's also lipid bilayers have a vast number of carbohydrate appendages. So off of these, this is a carbohydrate appendage. It's called a glycan. The artist is just showing us a few of these. A cell is covered with these. This is how cells recognize each other. How does one cell recognize another cell? By these carbohydrate assemblies that they recognize each other. They have this recognition patterns. And so you can tell one cell from another. And so if you just take nucleotides, if you just take DNA, DNA is so complex and it has so much information. No, if you have 6 A bases, A, how can you arrange those? What are the different ways you can arrange it? Just that one, A. That's it. One way you can arrange 6 bases. Look at carbohydrates. If you just take the carbohydrate D-pyranose, just a standard carbohydrate, just D-pyranose, you have 6 of those. Has over 1 trillion ways it can assemble. 1 trillion ways it can assemble just with 6 of them. These have much more than 6. And if it's not assembled right, guess what? The cell dies. You change any one of the carbohydrates, it results in cell death. There is much more information stored in carbohydrate assembly than in DNA. Yeah, much more information can be stored in carbohydrates. You want to build a massive computer? Build it based on carbohydrate assembly. Much better than DNA assembly. It's just a carbohydrate assembly is really hard to control. Nobody knows how to control this. Nobody. But somehow, on a prebiotic earth, with nobody around, under a rock, it figured this out. How do origin of life researchers address this problem? They don't. They don't. Next slide. Interactomes. This is the non-covalent interactions that function within a cell. Nobody knows how a viable cell emerges from massive combinatorial complexity of its molecular components. And of course, nobody's ever synthetically mimicked it. An interactome is the whole set of molecular interactions in a particular cell. If one merely considers protein-protein interaction combinations in just a single yeast cell, the result is an estimated 10 to the 79 billion combinations. That's estimated by these folks at Johns Hopkins and Brussels, all right? That's not my estimation. These are biophysicists, all right? So just to give you an idea of how big that number is, that's 10 to the 90th is the number of elemental particles in the universe. That's 10 to the 90th. This is 10 to the 79 billion. That's a big, big number. People do not understand numbers. They just don't. You say $1 million, $1 billion, what's the difference? $1 million or $1 billion? Well, let me put it in a way that you understand. 1 million seconds is 11 days. So you ask somebody, will you marry me? I'll tell you in 1 million seconds. Okay. If they say 1 billion seconds, that's 32 years. All right? You feel that now? 1 million to 1 billion? And then if they say 1 trillion seconds, that's 32,000 years. Huh? You see the difference when you go up 3 orders of magnitude? You go 1 million, 10 to the 6th, to 10 to the 9th, to 10 to the 12th. That's 10 to the 79 billion. That number's crazy big. That's just in a single yeast cell, the interactions between the non-bonded interactions. So how does the information flow? Information flows through non-bonded interactions through electrostatic potentials, which physicists call a virtual photon. Information goes down these at the speed of light. That depends on ordering between molecules, non-covalent interactions. These have to be all kind of assembled right. And that's why this information, you don't dehydrate cells and rehydrate them and get them to work properly. When a cell divides, it collapses down, puts the information on both halves, and brings it into both sides. So that that information keeps going to the next and the next generation. Because when you lose these intermolecular interactions that are non-covalent, non-attached, you've lost your information flow. Big problem. Nobody has explained. Nobody in Origin of Life ever mentions the word interactomes. Never will you hear it in their literature. Next slide. Proto-turkeys. Origin of life protocell assembly is akin to buying 20 pounds of sliced turkey meat, adding a gallon of turkey broth, warming, sticking a few feathers, and suggesting that a live turkey will eventually come gobbling out. If given enough time, or that a proto-turkey or extant turkey has been synthesized. This is exactly what is done in origin of life experiments. It's exactly what is done. If given enough time, a turkey's going to come out. And people buy this stuff. There's a whole area of research called Origin of Life research, and they've been doing this same thing since 1952. Next slide. Critical for life is the origin of information, DNA or RNA. The information is primary and the matter is secondary. We can't even get the matter, carbohydrates, nucleic acids, lipids, proteins, let alone the information. What is the code? Even if you had the nucleic acids and even if you could hook them up, what's the code? Where do you get that code? So we heard a little bit about this earlier in this conference. The information is primary. That's the information. It can be stored on all sorts of mediums. So I have an idea, and then I transcribe this onto a piece of paper, and now it's on the paper. And then I type this into my computer, and it goes into DRAM. And then when I hit save, it goes into flash memory. And then when I upload it to the web, that same information now is going through an RF wave into a box on the wall. And then from that box on the wall, it's going through a wire to some server farm someplace where it goes right back down into transistor-based flash memory. That same information, which was here in my hand to the paper, in my computer in several different forms, in an RF wave, same information, different medium. The information is primary. The medium can change all the time. So for example, the molecules in our bodies are always changing. It used to be said that the molecules in our bodies turn over or changed every 7 years. And that's a bunch of nonsense. They're changed continually. It's much less than 7 years. And some guys wrote, well, the molecules in your teeth aren't changed. And then I sent them, I did 1 second Google search. I mean, your teeth are constantly, you get dissolution, redeposition, constantly changing. Every molecule in your body is undergoing a proton exchange. Proton comes with a spin up, a proton comes with a spin down. Every one of these now is a different molecule in the sense that it has different atoms that make it up. Maybe the same pattern of atoms, but a different atom. You had a different proton exchange. Every molecule is continuing to change. We are dynamic structures constantly changing. What is the real me? I don't know what the real me is. If you want to say it's the matter is the real me, then you got a big problem because it's constantly changing. This is what we were talking about earlier, the difference between brain and mind. What is the real me? Next slide. You try to build a cell, even hypothetically, get the dream team, get the smartest people together. Can they build a living cell? I'll give you all the chemicals you want in homochiral form. And I'll give you even the informational code. I'll give you the whole code. In other words, you tell me how you want the DNA set up. I'll give it all to you. Now just assemble a cell. Go ahead. You got all your diacetyl lipids all in chiral form. Make your protocell. Make it, put in your peptides any way you want, set it up, get your carbohydrates out there. I'll even hook the carbohydrates together. You just tell me the pattern you want. Then you got to stick them on your cell. Can you do it? No, nobody can. Nobody's ever done it. Nobody can do it. That's not to say it won't ever be done. I'm just telling you, as of today, it hasn't been done, and it's far, far away from being able to do it. People will quote to me this, synthetic cells. Well, in 2010, Craig Venter's group copied an existing bacterial genome and transplanted it into another cell. So what happens? I buy, say, a Corvette. And so what do? I take the computer control box out of that Corvette, and I go to my clean room at the university, and I copy the chips. I copy them. And I put my chips that I copied into that control box, and I go back and I stick that in my Corvette. And I say, I built that Corvette. I made that Corvette. I did that. You just copied the same chip and you put it back in. That's all he did. He took another one in 2016, and he did something similar, but he took the control box and he knocked out all but 473 of the working devices, and he stuck it back in the cell. And like, whoa, you made a cell. No, he didn't. You just made a cell worse. You just chopped out a bunch of stuff and left just a few pieces to keep it operating enough. He didn't make the cell. There's all this complexity, all these interactomes. Nobody ever made that. Nobody knows how to do it. Next slide. So here's what's written in books that people read, people like you. What is life? I never read these books. You never see people like me read these books. This is the masses, but this is what they tell you. So here's this science writer, Regis. He says, life began with little bags of garbage, random assortments of molecules doing some crude kind of metabolism. That is stage one. The garbage bags grow and occasionally split into two, and the ones that grow and split fastest win. That's in his book on What is Life by Oxford University Press. Let me show you the primary literature. 2018 in the journal Nature. This is our top journal. You want to see what Nature will publish in this area? None of us could get away with this except Origin of Life researchers do. This guy is Nobel laureate Jack Szostak. Here's what he writes on how did life begin. Let's go through it. I'm interested in it. How did life begin? I mean, the guy's a Nobel Prize winner. He must know. The early atmosphere had no oxygen. It consisted mainly of nitrogen, carbon dioxide with smaller amounts of hydrogen, water, and methane. Lightning, asteroid impacts, UV light from the sun acted on the atmosphere to generate hydrogen cyanide, a compound of hydrogen, carbon, and nitrogen. Raining into volcanic or crater lakes, the cyanide reacted with iron, brought up water circulating through rocks. The resulting iron cyanide compounds accumulated over time, building up into a concentration stew of reactive chemicals. Okay, cool. Where are we? Life, as we know it, requires RNA. Some scientists believe that RNA emerged directly from these reactive chemicals, nudged along by dynamic forces in the environment. Huh? Nudged has no, you can't put nudged in any of our articles. No scientist knows what nudged means. They don't know. Nucleotides, the building blocks of RNA, eventually formed, then joined together to make strands of RNA. Some stages of the process are still not well understood. You think? How do these things hook together? Show me the chemistry. It's not there. Once RNA was made, some strands of it become enclosed within tiny vesicles formed by spontaneous assembly of fatty acid lipids into membranes, creating the first protocells. As the membranes incorporated more fatty acids, they grew and divided. At the same time, internal chemical reactions drove replication of encapsulated RNA. That is nonsense. Next slide. Here is, next slide, the figure that he showed. This is the figure from Nature. This is his figure. Cyanide derivatives and simple sugars. Those are not sugars. He says this is oxygen, carbon, nitrogen, phosphorus. That's fictitious. I don't know any sugars that have that chemical composition. You don't know that. He's lying to you. That's not real. Is this HCN? I mean, there should be a carbon, a nitrogen, and a hydrogen if that's HCN. And I'm colorblind, but I can see that's not really HCN. I don't know what that is. Maybe that's phosphate. I can see that. That'd be phosphorus, oxygen. Maybe that's phosphate. Okay. And then all of a sudden, boom, you got an RNA nucleotide. But the problem is that's not a nucleotide. It's the wrong structure. By far, that's the wrong structure. He didn't even put the right structure. At least he could have put the right structure. He didn't even put the right structure. Well, what's acting upon this to make it do this? Well, heat from here, UV light from there. Boom. Nobody knows. That's in Nature. That's not in an 8th grade textbook from 2018. People think I selectively choose these things from crazy places. No, this is Nature 2018. This is what confuses even professors. See this and they think, oh, yeah, people know. Nobody knows. This is a bunch of garbage.